Welcome to the Chapter 8 Part 2 lecture. You should use the information in this lecture to complete the Chapter 8 Part 2 guided notes, which of course you should complete before you attend class. Let's get started. So what comes to mind when you think of the term metabolism? For most people, metabolism is going to have something to do with either gaining or losing weight. Someone might say, "Oh, I can't lose weight at all. I have a slow metabolism." Well, while burning calories for energy is a part of metabolism, really, metabolism, biologically speaking, is the sum total of all the chemical reactions happening inside a cell. Now try to imagine that. It's a little bit difficult. There are literally hundreds or thousands of chemical reactions that can happen simultaneously inside a living cell. Here we have a metabolic map that shows just a few of the metabolic pathways that happen inside cells. So there's a lot going on. In this lecture, we're going to discuss how living cells organize their metabolic pathways, and we're also going to discuss how they use energy and how they use something called enzymes to help control which chemical reactions happen at which times. Cells organize their hundreds and thousands of chemical reactions into small systems called metabolic pathways. A metabolic pathway is a small set of chemical reactions, all of which are linked together. For instance, in this example, we have reactant A, and it's going to be changed into reactant B through one reaction. Then in a second reaction, it'll be changed into C, D, and finally E. At this point, from C, we could also go into another pathway and make F and G if that's what the cell requires. Examples of metabolic pathways include photosynthesis and cellular respiration, which we will get into in later chapters. Metabolic pathways consist of a series of chemical reactions, all linked together. At the beginning of any metabolic pathway, you're going to have the initial reactant. That's the ingredient that the cell has to work with. At the end of any metabolic pathway, you're going to have the end products. That's the product that the cell actually wants to make and to use for some purpose. Now, in order to get from A all the way over here to E and G, the cell has to go through a series of chemical reactions. So it has to change A into B, and then B into C, C into D, or F. All of these chemicals that are made in the middle are not the end product, so it's not what the cell wants to end up with, but they are still useful because they can be used to make other chemicals. We call these intermediate products. C, for instance, can be transformed into either D or F, so it's at a branching point in the metabolic pathway. How in the world do cells control these hundreds and thousands of reactions that are trying to go on simultaneously inside? Well, cells have three major means of controlling their metabolic pathways. First, they can control the flow of energy from one reaction to another by coupling them. Just to remind you, if you wanted to perform an endergonic reaction that requires energy, you would first have to perform an exergonic reaction, because exergonic reactions provide the energy for endergonic reactions. So if you have a metabolic pathway that is endergonic in nature, requiring energy, you can shut that entire pathway down by preventing the exergonic reactions that power it from happening. Another set of tools that cells have for controlling the energy flow between their reactions are these things called energy carrier molecules. Now, we haven't talked about energy carrier molecules much yet, but we will address them in the next chapter. The energy carrier molecules include things like NADH, and what these molecules do is they pick up energized electrons, energy, at one point in the cell, and then travel to another point in the cell and drop that energy off into another reaction. So these guys are like delivery trucks or dump trucks full of energy that deliver that energy where it needs to go. Now, if you can control the actions of these energy carrier molecules, you can control which reactions happen, where they happen, and when they happen. Finally, cells can use proteins known as enzymes to help control which reactions happen and when and where these reactions happen. Notice that in this example pathway, each step in the pathway is controlled by a specific enzyme, and they're all different enzymes. So imagine that this pathway is actually like an assembly line in a factory, and each step is controlled by a specific worker who has been trained to do that specific job. Now, if I wanted to shut down this entire pathway, really all I have to do is shut down the actions of enzyme number 1. Because of the rest of the pathway depends on the actions of enzyme 1, by shutting him down, I can shut down the rest of the pathway and prevent it from happening.
Enzymes.
Hey, Professor. Just the other day, I saw this small man swallow an entire steak bigger than the size of his head. And it got me to thinking, how could he possibly digest all that in just a few hours?
Well, the steak is first chewed in the mouth into bits and pieces that fall into the stomach. You might already know that the stomach contains a powerful acid that can dissolve even metals. But what you did not know is that this acid is not what ultimately breaks down the steak. No, it's very tiny and special molecules called enzymes that do the job. Strangely, these enzymes find the stomach acid a cozy place to work in.
Whoa! It's like small piranhas finishing up a whale in a sea of acid. Imagine what I could do with that!
Ooh. Huh?
Well, it doesn't work that way.
What do you mean it doesn't work that way?
I knew you were going to say that. To find out, you need to know what exactly enzymes are and how they work. Whenever you're ready, click Next to continue. What are enzymes? They are proteins that speed up chemical reactions. Without them, reactions can still occur, but they would be too slow to support life. There are many different types of enzymes, and they come in all manner of shapes and sizes. For example, what you call the piranha enzyme is actually pepsin. It is only found in the stomach and digests proteins. In your mouth there is amylase, which breaks down starch. In your cells alone are hundreds of enzymes that control your cell activity and defend them from invasions by microbes like bacteria and viruses. Though enzymes can appear very different, they all share a few characteristics in common. Let's now take a look at what these are. Number 1, each enzyme has an active site. This is the groove in the enzyme where the molecules they act on, called substrates, are captured and made to undergo a reaction. The substrates are either broken apart or combined together to form products. For instance, starch found in the bread that you eat is digested to form simple sugars called glucose that is then absorbed into the body. Glucose is used up as energy, but when there's too much of it, special enzymes combine the glucose molecules together to form a giant molecule called glycogen, which is then stored away in the liver and muscles for later use. Number 2, enzymes are very specific. Each enzyme can only bind to one type of substrate. Let's look at the case of lipase enzyme, which breaks down fats. Molecules like glucose and proteins cannot fit into the active site of lipase. Only fat cells can bind to the active site because they have the right shape. This ability to act on just fat cells is very important for lipase. Lipase converts fats into energy. Now imagine what would happen if lipase can also convert proteins into neurochemicals for the brain to function well. If you no longer need the extra energy from your storage of fats, lipase will be deactivated. But this means that lipase can no longer produce precious neurochemicals for your brain. In other words, when you stop burning fats, you end up damaging your brain. To prevent such freakish scenarios from happening in our body, enzymes must only act on one type of molecule. Number 3, enzymes are recycled. Enzymes are not altered by the reactions they speed up. Immediately after they finish one reaction, they can act on a new substrate. This way, only a small amount of enzyme is needed to speed up thousands of reactions.
Of the tools that cells have for controlling their metabolic pathways, enzymes are the most varied and probably the most important. Enzymes are a special class of proteins that help control chemical reactions. They act as catalysts. Now, catalysts are any chemical that helps speed up chemical reactions. You may have heard of non-biological catalysts. For instance, there are certain car parts that have catalysts inside, such as platinum, and the presence of those metals helps speed up the reactions going on inside the car. Enzymes are a biological catalyst, so they speed up chemical reactions inside living systems. Enzymes are capable of catalyzing lots of different kinds of chemical reactions. They can build molecules, they can also break molecules. For instance, here we see a big purple enzyme taking in this dimer, and it breaks it into individual monomers. Enzymes are only able to catalyze reactions that would normally happen by themselves. So given that, why are enzymes even necessary? Well, certain chemical reactions have to be performed almost constantly in order for living things to remain alive. If we stop performing those reactions, we basically die. Now think about what has to happen in order for a reaction to happen. The correct reactants have to hit together. They have to hit together with sufficient force to react, and they also have to be in the correct orientation when they hit together in order to react. If we were to sit around waiting for this to happen spontaneously, we'd die while we were waiting. So enzymes are necessary to speed up those reactions, to make sure that they happen at the correct rate so that we can still keep breathing. This chart shows the rate of a particular reaction without the enzyme and with the enzyme. And what you can see is that especially initially, the rate of reaction with the enzyme is much, much faster, much steeper than the spontaneous reaction without the enzyme. Enzymes speed up reactions in two ways. First, let's consider what they physically do to the molecules they work on, the substrates. And I have a really stupid analogy to help you understand this. Let's say that you have two friends that you think would make an amazing couple, but they don't know each other. So you decide you're going to play matchmaker and you're going to throw a party. So you throw this party and you invite both of them. Well, that's great, they're both there, but they're on opposite sides of the room, not talking. So maybe you get one of them and take them over to the other. Well, that's better, but now maybe one of them's facing a wall for some reason. I didn't say they were very bright. All right, so you get the other person, you turn them around so they're facing each other. Maybe you give them both some punch or you tell a little joke and kind of get the conversation going, get the reaction started, right? Well, that's essentially what enzymes do. They gather up the reactants for a particular reaction and they physically bring them close enough together to react. By sticking to the enzyme, the substrates, the chemicals they work on, are also ensured of being in the right orientation, so they're facing each other in the right way to react. So enzymes physically bring the chemicals together. Enzymes also help catalyze reactions by lowering the amount of activation energy needed to get that reaction started. Let's examine this on this chart. Here we have the progression of a reaction without the enzyme. So you notice that this is exergonic because the reactants have more energy than the products. But the reaction will start, and then we have this big bump. That's the activation energy. That's the energy that has to be taken in order to get the reaction going. And then once that's met, the reaction can proceed down here to the products. Next, let's look at the reaction with an enzyme included. Because the enzyme has done all this legwork, because it's gone out and gathered up the reactants and put them in the right orientation, the reaction itself is going to require a lot less energy to get started. It's not just depending on those reactants bumping together with sufficient speed anymore, so less energy is required. So the energy of activation here, this bump, is much smaller. And then you notice that it can proceed to the same products. So enzymes help catalyze and speed up reactions because the reactions with the enzymes require a lot less kinetic energy. Enzymes that work in metabolic pathways are a lot like factory workers that work on assembly lines. Those types of workers are typically trained to do one or two very specific jobs, and they do those jobs over and over all day. In the same way, enzymes are specific for the molecules they will work on. In other words, they only catalyze very specific chemical reactions, and they only work on specific molecules that we call substrates. Now, why is this? Well, remember that enzymes are proteins. Proteins have a very specific three-dimensional shape, and these enzymes are going to have a groove in them somewhere, a little kind of divot or hole, called the active site. Only specific substrates can fit into specific active sites. In other words, this little substrate has the right shape to fit in that active site. If another molecule comes along and tries to fit in the active site, it's not going to fit. So the enzyme can only work on molecules that will fit in its active site. Now, why would a cell want to have thousands of different enzymes with each one only being trained to do one specific job? That seems like overly specific training. Well, think of it this way. If you have metabolic pathways consisting of thousands of different reactions, and you need to control each reaction, you really only want one enzyme that can catalyze each reaction. That way you can control each specific reaction by controlling the actions of that specific enzyme. So it gives the cell more control over specific reactions. Enzymes are a type of protein, and something we learned back in chapter 5 is that proteins have very specific three-dimensional shapes, or conformations. Now, one of the neat things that enzymes can do is change their conformation, change their shape, and that allows them to catalyze the reaction. For instance, as this enzyme is taking in this substrate, it might change the shape of its active site just a little bit so that it can get a better grip on that substrate. The steps that an enzyme goes through in catalyzing a reaction is known as the enzymatic cycle. Let's look at the steps. First, the enzyme is going to take the substrate in and fit it into its active site, and it's going to change its conformation a little bit to get a really good grip on it. At this point, we say that the enzyme substrate complex has formed. Here you can see it. Next, the enzyme is going to change its conformation again to make the reaction happen. In this case, we're taking two little monomer subunits and putting them together into a dimer. Next, the enzyme has to change its shape again in order to release the product. So the product kind of pops out of the enzyme. Now at this point, the enzyme has not been affected by the reaction. It can now go out and pick up another substrate again and do this again and again and again, hundreds or even thousands of times. That's why we refer to this process as a cycle. Enzymes are really excellent little workers. Let me give you a few examples of some well-known enzymes. The first is known as DNA polymerase. In order for a cell to reproduce, it has to make a copy of itself. And in order for it to do that, it first has to copy all of the DNA inside the cell, all of the instructions on how to be a cell. We call that process DNA replication, and DNA polymerase plays a key role in DNA replication. Let's take a look at it. Here we can see that we have an old strand of DNA known as a template strand. And here's DNA polymerase, and what it's doing is it's adding new nucleotides onto the old strand. So it's matching a new strand to an old strand. Now, there are a number of enzymes that actually participate in this process. It's not just DNA polymerase, and we'll definitely take a look at that in a later chapter. But for now, you can just know that DNA polymerase helps copy DNA. Many different types of DNA polymerases exist inside cells. One polymerase specifically to be aware of is this guy, Taq polymerase. The Taq in Taq polymerase stands for Thermus aquaticus. That's the genus and species name of the bacterium from which this enzyme was originally harvested. Thermus aquaticus lives in hot springs. So for instance, here's some hot springs in Yellowstone Park, and you can see that it's a pretty inhospitable environment for life. Yet life is found there. Because this thing lives in a very high temperature environment, it means that all of its enzymes and all of its structures have to be well adapted to working at high temperatures. So the enzyme Taq polymerase is thermostable. That means that we can heat it up without it denaturing. And that makes this enzyme very useful in a process known as PCR. Now you're going to learn about PCR in class. It's a method for causing artificial DNA replication inside a test tube. And when we do something inside a test tube, we call it in vitro. But through this process, we can take a single little strand of DNA and we can make lots and lots and lots of copies of it. We can replicate that piece of DNA as many times as we want so we can study it and manipulate it and do interesting things with it. And that wouldn't be possible without this enzyme Taq polymerase. If we took DNA polymerase from a human, for instance, and tried to use it in this process, the temperatures involved in PCR would pull it apart and denature it. So we owe a lot to Taq polymerase. It's a very useful enzyme. Another type of enzyme that's used very commonly in the molecular biology lab are these guys known as restriction enzymes. Restriction enzymes are like molecular scalpels or molecular cutting tools, and what they cut specifically is DNA. Now let's think about that. Why in the world, as a living cell, would you want to have enzymes present that can cut up your DNA? Well, these enzymes are produced in prokaryotic cells, so things like bacteria and archaeans, and these cells actually make them to fend off viruses. Remember that viruses will invade cells. Viruses try to put their genetic material inside of living cells and turn those cells into virus factories. So what these cells use these enzymes for is to cut up the viral DNA as it's invading, and it helps protect them from viruses. Now we can use these enzymes in the lab as well. Let's say we want to take these two pieces of DNA and we want to combine them into one. Maybe we're taking a gene from a bacterium and combining it with a gene from a plant or something like that to make a genetically modified organism, or for some other purpose in the lab. Well, what we can do is take the DNA from both sources, we can snip it at very specific places, and these enzymes will snip DNA down to individual base pairs that you want to cut. So it separates them all the way through. And then we can use another enzyme called ligase to actually glue them together. And we end up with this stuff called recombinant DNA. So restriction enzymes are used very commonly to help us manipulate DNA in the lab. Another very useful enzyme.